Welcome to Biology Tutorial with Teacher Joy. Today, we will learn the use of Punnett Square in genetics. So, sit back, listen to me, eyes on me. Are you ready? Then let's go. Let's talk about the Punnett Square. A Punnett Square is a diagram composed of boxes, and it was invented by Reginald Punnett. In predicting the results of monohybrid cross, we can use the Punnett Square Method. Punnett Square Method is a diagramming technique for predicting the possible outcome of a mating between individuals. Now, let's perform a monohybrid cross. Now, what is a monohybrid cross? Yes, it is a cross involving one trait of contrasting versions of an individual's characteristics. For the purpose of demonstration, let me use the characteristic of eye color. Though studies show that eye color does not confirm Mendel's complete dominance. But for this lesson, we will use the eye color to demonstrate the use of the Punnett Square Method. For an instance, one eye color is dominant over the other eye color in a certain human population. A person may either have brown eyes or dark eyes. Brown eyes or dark eyes are what we call the phenotypes. Phenotypes are the observed traits in an individual which are manifested in their physical appearance. Let us assign small b for brown eyes and big B for black eyes. Small b and big B are the genotypes. Genotypes are the genes on the chromosome that control the phenotype of a person or the observed physical appearance of an individual. The big B allele is the dominant allele. Dominant allele is the version of the allele that is always expressed phenotypically in an individual. The small b is the allele that is called recessive. Recessive allele is the version of the allele that is masked by the dominant allele. Let's have a sample scenario. Let's start with step number 1. Assign a symbol for each allele. In this example, our symbol for the allele of the eye color is B. Small letter b for the brown allele and big B for the black allele. Next, we have to identify the phenotype in all possible genotypes. The possible genotypes are homozygous black and homozygous brown. Homozygous is a genotype with only one version of allele. In other words, it is a purebred. On the other hand, heterozygous is a genotype with two versions of allele. In other words, hybrid. Phenotypically, Homozygous and heterozygous are either seen as black eyes or brown eyes. Step number 2. Determine the genotype of each parent and indicate a mating. Now let us assume that both parents are heterozygous for the trait of eye color. This means that the father has an allele for black eyes and an allele for brown eyes, which is the same in the case of the mother. The mother, just like the father, has the allele for the black eyes and the allele for the brown eyes. Phenotypically, both of these parents have black eyes. From the results of step number 2, let's determine the possible gametes or gametes each parent can produce and write the symbols for allele in the Punnett square. In a monohybrid cross, the Punnett square consists of 9 boxes. Let the y-axis be the gametes or gametes for the father, and let the x-axis be the gametes or gametes for the mother. Now, what are the possible gametes or gametes for the mother and the father? That is a great job! The mother has an allele for the black and an allele for the brown. Likewise, the father has also the allele for the black and the allele for the brown. So we have now our step number 3. For step number 4, let us determine all the gene combinations that can result when these gametes or gametes unite. Let's fill in the Punnett square. Big B cross with big B equals to big B. Small b cross with big B equals big B and small b. Big B cross with small b equals big B and small b. Small b cross with small b equals 2 small b's. Do you have the correct answers? That is another great job. So, we have our possible gene combinations that can result when the gametes of the father and the mother united. And finally, step number 5. Determine the genotypic and phenotypic ratio of each of the gene combination. From the Punnett square in step number 4, let us list our gene combinations. The first combination is homozygous black. How many did we get? Yes, 1. That is great job. Next combination is a heterozygous black eyes. How many did we get? That is correct. 2. Good job. And the last combination is a homozygous brown eyes. And how many did we get? That's 1. That is great job. Therefore, our genotypic ratio is 1:2:1. How many of the offsprings will have black eyes? Very good. 3. And how many of the offspring will have brown eyes? Yes, that is great job. Only 1. So we have 3:1 phenotypically. We get now our phenotypic and genotypic ratio. Our genotypic ratio is 1:2:1. And our phenotypic ratio is 3:1. Now that we're done with the Punnett Square Method, we can predict that out of 4 children, 3 may have black eyes and only 1 may have brown eyes. See? Genetics is easy. I hope that you learned a lot of things from me about Punnett Square Method. See you in the next lesson tutorial. This is Teacher Joy signing out.